Hello, everyone. Thank you to be here today for this live session. We will wait a few minutes to allow all people to join us, and we will start the presentation. So here you can see the camera of our expert, Xavier, our speaker, and Erika. They will be here to answer all your questions on our delivery platform. So let's wait 1 minute and we will start the presentation. Okay, perfect. That's time. So let's go. So again, hello everyone, and thank you for joining us today. So we are thrilled to have you here for this webinar. So over the next 45 minutes, we will explore the world of drug delivery with a special focus on lipid nanoparticles. While LNPs have revolutionized drug delivery, researchers you can often face significant challenges in LNP preparation, analysis, and most importantly, designing them for precise delivery to specific cell type, to your model. So through this presentation and a dedicated case study, we invite you to discover how our Europe-based contract research services laboratory, led by a team of experts, understand these challenges and can support you in overcoming them from formulation to targeted delivery. So we really hope this session will provide you valuable insight into how our expertise and services can enhance your research. Okay, but first, let me introduce the expert joining us today. So Xavier Warnet, our speaker, is a project manager in our lab specializing in delivery and formulation screening project. So during the Q&A session, he will be joined by Dr. Erika Siri, one of our lab experts who work on RNA production and delivery project. And I am Frédéric Samazan, your host and moderator for this session. But before we begin, I would like to go over a few housekeeping guidelines for this session. First, this session is being recorded, and we will share the recording with all of you after the webinar. If you have any questions during the presentation, please use the Q&A box located at the bottom of your screen, and we will address them during the dedicated Q&A session at the end. Also, please take a moment to complete the survey that will appear in your browser when you will leave the session. Your feedback is invaluable, and if you would like more information, let us know. We'd love to follow up with you. Okay, a quick overview of this session. I will begin with an introduction of Tebubio and of course our speaker today. Through this session, Xavier will address some LNP basics, key challenges in delivery and formulation. He will provide insight into Tebubio's delivery platform and showcase our capabilities through a dedicated case study. Additionally, we will highlight how our collaboration with innovative partners are driving advancement in vivo drug delivery. And of course, stay with us until the end for the live Q&A session where our expert will answer all your questions. Okay, quick word about Tebubio. So we are a pan-European company, and since more than 70 years, our objective is to support life science researchers on a daily basis and facilitate your project thanks to our unique positioning and holistic range of solutions. Our European coverage gives you an easy ordering to a broad range of innovative solutions sourced from trusted and ethical suppliers. If you are short on time or need specialized expertise, you can outsource part of all your project to our contract research service lab in Europe. They are covering everything from mRNA production and formulation, our focus today, to in vitro model development and target identification. We also take the SL out of ordering and logistic with our easy procurement system backed by years of experience. So finally, to conclude on that part, with Tebubio, you can focus on your research. We will handle the rest. I will go a little bit deeper into to introduce you our laboratory platform. So our lab has been growing for over 20 years. It's composed of scientific experts across various fields and a team of technicians. And we offer a wide range of services. Our expertise is structured around 4 key platforms you can see at the screen. The first one is the RNA-based therapeutic discovery, supporting RNA production, delivery, and analysis of the expression. The second one is in vitro modeling for the culturing and optimization of your experimental model. And the 2 last one are biomarker mapping and data analysis, providing comprehensive insight into your research. So, of course, you know, but today we will focus on our RNA platform, specifically highlighting our capability in delivery and formulation screening. But, okay, without further delay, let me introduce our today's speaker, Dr. Xavier Warnet. Xavier joined Tebubio 1 year ago, in February 2024, as a project manager on the RNA-based therapeutic discovery platform. With a robust academic background, including a PhD in biochemistry and biophysics, he brings deep expertise in biomolecular research, analytical techniques, and innovative drug delivery technologies. Prior to his role at Tebubio, Xavier led the diagnostic team at LPS Biosciences, focusing his job on the structural and functional characterization of lipopolysaccharides through advanced analytical technique. Now at Tebubio, Xavier leverages his expertise to guide clients in optimizing their delivery project. And of course, he continued to develop and expand Tebubio LNP Delivery Platform. But with that, I will hand it over to Xavier. So thank you again for joining us. And we hope you will enjoy this session.
Okay, so good morning, everyone. Thanks, Frédéric, for the nice introduction, and thank you, everyone, for joining. So, as Frédéric mentioned, I will go through the webinar now, starting with some basics around the LNPs. Okay, so as you all know, LNPs are approved by the FDA for mRNA delivery. They are biocompatible by nature because of their lipid-based composition. They are quote-unquote easy to synthesize. I mean, it's not that easy, but it's easy in the sense that chemistry of lipids has been known for decades through all the work that has been done on biological membranes, biomimetics like liposomes, SUVs, and stuff like that. And the good thing is that LNPs are quite versatile too, because they allow you to encapsulate different cargos centered around nucleic acids, meaning they can encapsulate mRNA, microRNA, sRNA, or even DNA. If you tweak a little bit, it's a bit more difficult to encapsulate DNA, but it's possible. So as you probably all know, LNPs are composed of 4 different classes of lipid, or at least 4. You have the 2 main ones from a molar standpoint are the ionizable or cationic lipids, which represent roughly half of the molar ratio of the lipids presence. So the ionizable lipids are crucial for the LNP because they are the one that dictates a lot of properties, meaning they are the one responsible for mRNA encapsulation, but they have also an important role in endocytosis and membrane and cellular uptake of the LNPs, and then endocytosis, and which leads to the mRNA translation, basically. Regarding the sterol, so that's the second highest component from a molar ratio again. It's also very important for the structure of the LNPs, but it has also a role in the uptake because it allows some serum protein to bind to the LNPs, and that favors the uptake of the LNPs by the cells. Then we have some structural lipids. It can be different kinds of lipids. You've probably heard of DSPC or DOPE as structural lipids. They also provide some structure to the LNP. They influence the biophysical characteristic of the LNPs, and they can also have an impact in the endocytosis procedure. And finally, it's quite an important part, the PEG lipids. So it's very few molar, but it's really crucial because it really dictates the size of the LNPs. And as you all know, sizes of LNPs are really important, especially in vivo for crossing several blood barriers. But they also have an important role in controlling or I would say tuning the biodistribution and also the circulation time of the LNPs in the blood. So now on to the challenges. So here are some examples of challenges that we encountered or that we think that you might encounter during the LNP preparation. So I've divided in three categories. So you have mRNA preparation, lipid composition and mixing, and LNP formation. So regarding the mRNA preparation, obviously mRNA is crucial for your application. So you have to have proper sequence. What I mean by that is, so you have to have a proper coding sequence, obviously with optimized codon, that's for sure. But you also have to have optimized UTRs, depending on for example, on the application you want, on the cell type you're looking at. You also have to have poly-A. That's also important because if not, then you would have very low translation and expression of your protein. So this part is really crucial. Then you have also what I call the modification of the mRNA, which basically covers the capping and the modified UTPs. Both are very important because capping is essential for translation. And modified UTPs are essential because, as you might know, UTP is quite immunogenic. So if you don't have modified UTP, then you will have quite toxic mRNA, and then you would have poor translation. That's the same idea. And then this is the last one is quite a bit more technical and might sound trivial, but the pH of the mRNA solution is actually very important because, as you know, so first you prepare the mRNA and then your lipid mix, and then you mix the two, and that forms LNP in a nutshell. But you have to be at a pH that is below the pK of your ionizable lipid for that to work. So you have to know the pK of your ionizable lipid and then be way below that. So that's something very important because if not, then the encapsulation will not work. Basically, you will have very poor encapsulation efficiency and your system will not work. Then into the second category, so the lipid composition and mixing. So obviously the type of ionizable lipid that you are going to use, this is the cornerstone basically of the formulation, right? So you have to select the right ionizable lipid for your application. And this depends on your applications. Obviously, there are really a huge development in ionizable lipid synthesis. So you can have access to, I don't know, hundreds of ionizable lipids. So it's a bit difficult to find the right lipid, but it's also crucial for you to really think about that. And following that, the proportions between all the components I mentioned before are also important because they will influence either the biophysical characteristic of the LNPs, the size, the polydispacity, but also how the encapsulation will work, the potency of encapsulation, and then also downstream the encapsulation, the endocytosis, sorry, and then mRNA translation. So those two are really crucial. And then if we go more into the mixing part, the proportion of lipids to mRNA, the famous N2P ratio, is also crucial. It's a bit more like I don't know how to say it in English, but you have to do experiments to really optimize this N2P ratio, right? There are some basic, like you have to be around 6 for it to work most of the time, but you really have to optimize around that because it also affects the efficiency of the transfection and translation of your mRNA. And then into the mixing. So this is also important. So basically you can do LNPs by hand, but usually you get very low encapsulation and efficiency. And since you have very low control regarding encapsulation, then the risk is that you will have poor reproducibility. So that's why there's a lot of work that has been done in developing new microfluidics devices that I will introduce some later on, that really helps to control all the process and also allows you to tune the LNPs so that they have particular size, particular polydispersity index, and so on. So it's really a nice way to really shape your LNP the way you want, basically. And then to the next part, the LNP formation. So I put just 2 points here, just to emphasize that LNP stability remains an issue. You can add cryoprotectants. There are even some possibility of lyophilization, even though this one is it's a bit more difficult. But even with cryoprotectants, you have to bear in mind that LNPs and mRNA are by nature not stable. And because even if you freeze, I mean, if you freeze lipids, usually it's not good because they tend to fuse after thawing, and you lose the structure of your LNP. So just bear in mind that. And as I mentioned, so LNP size and monodispersity or homogeneity is really crucial, because especially in vivo. In vitro, you will see it's less a problem, but in vivo, it's really crucial because it will allow your LNP to cross, as I mentioned, several barriers, and also to be uptaken by the cells. So now onto a little bit of overview of our delivery platform. So I just put the mRNA workflow that we have. So we can start by doing mini to mid-scale production of your mRNA, either using our proprietary vectors that has been optimized for mammalian cell expression, or your vectors. It doesn't really matter. So what I mean by mini to mid-scale, it's between 100 micrograms to a few milligrams, basically. Then there is the delivery part, which is the focus of this webinar. And it's pretty much the same scale. It's between few micrograms up to a few milligrams. And then we can also work with your mRNA if that's something of interest for optimizing formulation. And then we can also do some downstream analysis if you're looking into in vitro transfection or looking into biomarkers and so on. And if you're interested in upscaling to GMP, we also have partners for both the mRNA part and the LNP also. Yeah, and if you're interested, as Frederic mentioned, you can have a recording of this webinar, and we are always available should you have any questions. Yeah, so what we have in the lab right now, so we have a Spark Nano Assembler from Precision Nanosystem, which is a very nice tool if you're looking into formulation screening, because you use very little amount of mRNA. It's like between 5 to 20 micrograms basically per cartridges. And also you use a low amount of lipids, so that's quite nice. And end of last year, very end of last year, we acquired a new instrument, which is called the Tamara from Insight Therapeutics. And this is a very nice instrument because it allows us to upscale quite a bit now. So with that, you can encapsulate milligrams of mRNA. And as I mentioned earlier, so it's a microfluidic system, so where we control a lot more parameters than with the Spark. So we can really finely tune the LNPs and achieve results that we can't really with the Spark, like regarding the size, for example, of the LNPs. So this is very nice to have. And yeah, and now into what we can do for you. So this is just an example from a customer inquiry. Sorry. So what he wanted to do was to identify potential LNPs formulation for targeting cancer cell models, HCT 116, and he had some trouble finding the adapted lipids and the formulation parameters, and also all the QCs around that, meaning both the biophysical characterization of the LNPs, like the size, the polydispersity index, but also the in vitro transfection efficiency. So what we built together with the customer was to design 15 different formulations. And then we performed small-scale screening using the Spark instrument that I mentioned earlier. And we qualified this formulation based on their biophysical properties. using DLS and the quantification of encapsulated mRNA using the ribogreen assay. And then we performed in vitro transfection just to assess the efficiency of this formulation. So the 15 different formulations that were built were based on 5 different ionizable or cationic lipids. So we had 2 classic one, the LC0315 of in the Pfizer vaccine and the SM102 from the Moderna vaccine. Then we have one of the lipid brick. So I will go a little bit more onto the lipid brick later on. But this is a cationic lipid that is quite versatile and that can be used on its own or with other ionizable lipid. And we coupled it with the DODMA, another cationic lipids, which has been shown to be quite efficient, the mix of the two. And then we also included an ionizable lipid from one of our latest suppliers, Certest, the CPLC 0741, because they Certest really showed some very nice in vivo data from this lipid. So we decided to test this. And we also use different structural lipids because we've seen, and it has been shown in the literature, that depending on if you use DSPC and DOPE, you could have different encapsulation endocytosis efficiency, basically. And also to help us track the LNPs, we used custom-made fluorescent lipids, which is the Cyp3 conjugated, which was made by Echelon Biosciences, one of our suppliers. And we encapsulated between 5 to 15 microgram of mRNA. And we used, to control this formulation, we used EGFP that we made in the lab, which was Cap1 and 120 poly-A, and then also with N1 methyl pseudouridine. So we pay from the usual quality control like DLS and ribogreen assay. So you can see the results for all 15 formulations. So I should mention here that we did not optimize the formulation parameters for each of them. So we expected using, we use like all the same parameters for everyone. It was N2P ratio of 6, flow rate ratio of 3, and that was it. So we expected to have some formulation that had bad QC, so to say. For example, if you take the number 2, 3, and 7, they have very low encapsulation efficiency, meaning that we should work. If those formulation will look nice in vitro, we have to work around the formulation parameters to improve that. And similarly, if you look at formulation 9 and 10, they are quite heterogeneous. Right? The polydispersity index is quite high. And yeah, I forgot to mention, but the red box corresponds to the fluorescently labeled LNPs. So even though the QC did not look so good for some of them, we agreed with the customer that we should still go on into the in vitro because we know that for in vitro studies, it's not as important for in vivo. And if one of those formulation looks promising, then we can go back and optimize the formulation. But for rough screening, we thought it would be fine as it is. So for cellular transfection, so we use HCT 116, a cell in 2D models. So as you can see here, so those 8 are the one without any fluorescent lipids, sorry. You can see that depending on the formulation, you have quite differences in translation efficiency. So you can clearly see that the formulation number 4, 5, and 7, and 8 work quite well. Even though the 7 one had very low encapsulation efficiency, which means that if we optimize this one, it could be a really interesting one to use. Oh, I forgot to mention. So I anonymized all the formulation because I don't want to emphasize one lipid against the other one. The take-home message here is that there is not one lipid that is best. It's just that you have to find the one that fits your need, basically. So that's the reason. Next, we evaluated the fluorescent LNPs. So you can see here, so the red signal is the correspond to the fluorescent lipids, sorry. So this actually was quite helpful for us because you can really track your LNPs and see that basically for all of them, you have some, I don't know if they are endocytosis, but there is some interaction with the membrane for sure. but that doesn't correlate with protein expression. So we don't know what is going on because we don't have access. We haven't tested for that, but it's very likely that even though you have interaction with the membrane, it doesn't lead to endocytosis, or if it leads to endocytosis, there's no release of the mRNA into the cytoplasm, which is very interesting to actually see. So obviously you can see that similar to the non-fluorescent lipids, we clearly have some formulation that sticks out, like the 12, 14, and 15, basically. Even though you can see that, for example, the 14 and 15 lead to some cellular death. So that's also something to take into account when you are analyzing the results, obviously. So based on all these tests, so as I mentioned, so adding the fluorescent lipid really helped us to assess the LNP's formulation and to see what was the best one, basically. From this 15 formulation, we selected 3 of them, so number 4, 5, and 12. You can see here the summary of the parameters. So you can see that, for example, for number 4, we would have to work a bit around the encapsulation efficiency, so go back to the formulation parameters and optimize that. And for the number 5, it will be around the polydispecific index because it's a bit high. So the idea now is to test this formulation on a 3D model of these HCT116 cells. So this is what we are currently doing. I just show a picture here. So from formulation 4, I think. So it's worked already nicely. And so here because we had access to a very large library of lipid, we are really able to help our customer. And we offer the further really integrative approach, meaning that we work from mRNA up to the LNP and then to the cellular assay. And now the idea is to work with the customer mRNA, just to using the same formulation and just to see the effect of this mRNA on the spheroids. And then the formulation that we have built, they also can constitute a cornerstone for in vivo experiments, should that be the way to go. It would require, obviously, some optimization, but at least there's some basics to work with. Yeah, so as I mentioned, so we received the Tamara instrument from Insight Therapeutics end of last year, so it's still under validation, but we've performed some pretty promising tests. So here I just show what we can do also around formulation optimization, because so far I only spoke about lipids, but here I just show parameters that are known to influence either the shape of the LNPs or their efficiency in vitro or in vivo. So here we tested different flow rate ratios, N2P ratios, and total flow rate. So you can see that, for example, the flow rate ratio has quite a dramatic impact. So we for this one, yeah, I forgot to mention, we used the off-the-shelf formulation from ABP Biosciences that mimics the one from Pfizer vaccine, basically. So you can see that with this formulation, a flow rate ratio of 4 would be best, and an N2P ratio of either 4 or 10. So it's between the 6 doesn't work as well. So it has to be done again just to check. And the total flow rate does not affect so much the transfection efficiency, but it has an impact on the size, which is expected, even though the size is still quite large for these particles. But this is also something that we can work around. Okay, now onto our suppliers. So because they are an important part of what we do, so we have a network of high quality partners that allows us to have solutions for quite a few problematics that you could have. So whether you're interested in vitro validation, ex vivo studies, in vivo targeting, or mRNA-based vaccines. So regarding the in vitro validation targeted delivery, so thanks to Echelon Biosciences and Broadfarm, we have access to a very large library of either the ionizable lipids or cationic lipids from the literature. Most of them you can find there. They have also quite a large library of sterols, because I only spoke about cholesterol, but you can use a lot of different sterols that can also improve the transfection efficiency. They have also different pegylated lipids, because pegylated lipids is also important for controlling the size and also the circulation time, as I mentioned. They have also activated lipid through NHS, maleamide, or click chemistry if you're interested for conjugating whatever you want to conjugate basically with the lipids. And they have also like already modified lipids, fluorescent lipids, among other things. And then I also present one of our partners, Polyplus, which is now part of Sartorius, and their lipid brick chemistry. So here I will just speak a little bit about that. So the lipid brick is a library of 8 different lipids, so that you can see here on this table. And this is a very versatile tool because it can be used on its own to form LNPs, but it can also be used in combination with any other ionizable or cationic lipids to enhance the effect of this ionizable lipid. So that is very nice. And as you can see here, depending on the lipid brick that you use, you will have different efficiency in different cell lines. So that goes back to what I was saying at some point when I said that you have to choose the right ionizable lipid for your application, basically. So this is also a very nice tool to have. Then in ex vivo studies, so we have access to off-the-shelf formulation, for example, from Precision Nanosystem for doing CAR T cells. And here I present the results from Polyplus. So here they use their lipid brick technology and compared it to LC0315 LNPs. And you can see that the percentage of expression of the CAR is much higher than for the LC0315, while keeping the cells alive, which is a plus. Then for the mRNA-based vaccines, so we have access, as I mentioned, to off-the-shelf formulation, which are ready to use, that mimics the one from Pfizer and Moderna, so with the ALC0315 and SM102. But we have also access to other innovative solutions for doing mRNA-based vaccines, and that they have been validated in vivo, and they are both efficient and safe. Sorry, so that's what you can see here with the lipid brick. So it works the same as some gold standard in the mRNA-based vaccine. And then through our new supplier Certest, we have also access to a large library of ionizable lipid that have been shown to work very nicely compared to the gold standard. So here you see a viral quantifications, and you see that basically, so in orange, you have a gold standard mRNA-based vaccine, and in blue, you have LNPs using the Certest ionizable lipid. So they work very nicely, even better than the gold standard. And for the in vivo targeting, so again, lipid bricks, they have shown that they can bypass to some extent the liver. So that's what you can see here. So you have on-patrol LNPs on gray, and in yellow, you have the lipid brick LNPs. So you see clearly that the expression in the liver of the probably luciferase is way lower when you use the lipid brick, and it goes mostly in the lung and the spleen. And then Certest, so I will be a bit longer on this one. So Certest has developed a lot of different ionizable lipids. And the good thing is that they have been all validated in vivo first. So we know that all the lipids, they work in vivo, and in fact, they have designed some ionizable lipids that have some tropism for different organs. So for example, you see here that lipid 1 will go mostly into the lung. Well, yeah, mostly into the lung. Lipid 3 will go mostly into the spleen, whereas lipid 6 will go mostly into the liver. So it's very good because you really have some tropism, and your NP doesn't get too diluted compared to other formulations. So this is a very nice supplier to have for LNP design. So basically, so I hope I convinced you that we can really help you through your project by providing different answers to your problematics, whether it is which type of lipids, what experiments to perform. We are quite flexible around nucleic acid encapsulation. We can work with mRNA, we can work with oligonucleotides. We have access to a lot of different, as I mentioned, supplier partners that can really also help us and then help you finding the right solution for your problematic, basically. And I think that's it for me.
Perfect. Thanks a lot, Xavier, for this very clear presentation on main LNP problematic, our capability at Tebubio laboratory, and also the strengths of our partner, what they can provide us in terms of innovation and innovative lipids. So okay, perfect. So before the live Q&A session, I just would like to take this opportunity to present our next webinar session in this delivery series organized with our partner Certest, which will take place on Monday, 10th of March. So you can join us for an exclusive webinar where we will unveil the latest advancements in vivo drug delivery with Certest smart lipids and smarter targeting. During this session, Dr. Javier Jimenez Warren, CTO at Certest, will present key insights and findings from their latest research, showcasing how Certest proprietary lipids technology is transforming efficient and targeted in vivo drug delivery. So really, I really recommend you to attend this session and don't miss the opportunity to explore this innovative solution. And finally, how this collaboration between Tebubio and Certest allow a seamless transition from discovery, what Xavier just introduced you, to in vivo delivery, what Dr. Javier Jimenez will show you next month. Okay, of course, we will send you the registration link in the follow-up email. To wrap up our session, now let's move to the live Q&A session. Really take this opportunity to ask any question to our expert Xavier and Erika. Really feel free to share all your questions. So we already have some questions in the pipeline. We still have time, so don't hesitate to ask all your questions using the Q&A box. Okay, perfect. Let's move on the first question. What is the maximum size, for example, 40 kilobytes of DNA that could be packed within the LNPs? Can you give specific example of successfully LNP encapsulated DNA?
That's a very good question. 40K is a lot. I know that you can encapsulate siRNA, so it's already quite a big RNA. So 40, I'm not sure. You can also encapsulate, plasmids. So I guess the way I see LNPs is that they are a bit like nanodiscs if you've worked with membrane proteins. So basically, the lipids will accommodate around your DNA or RNA. So depending on I think what will mainly matter might be the structure of your mRNA. So if it's 40K, but it's somewhat, I don't know how to say that, but small in its hydrodynamic radius, maybe the particles will not be that big. So I don't have specific example aside of the siRNA and plasmids that can be encapsulated, but I think it's worth testing for sure.
If I can add one thing, so it's the encapsulation of DNA, it's something that our partners have explored. So I invite you to fill the form at the end of the seminar with like the specificities of your project, and we can discuss with them and get back to you with what we can provide as a solution.
Thanks for the question and your two answer. Perfect. Next question. Sorry. In the in vitro studies with a dose of 200 nanogram of RNA, did you report any toxicity of LNPs?
It depends on the formulation, but the answer is yes. And actually, if you transfect some empty LNPs without mRNA into cells, they can be toxic. But bear in mind that it's in vitro, and that means it's different mechanism than in vivo, where in vivo you have a lot of more mechanism that will prevent that. But I mean, LNPs have some sort of adjuvant effect, so they sort of also, make the immune system tilt a bit, right? So But yes, we did.
Okay, thank you. New question: do you have proprietary lipids?
Us, no, but with our suppliers, yes. So, for example, Satis that Frédéric mentioned, they have proprietary lipids. Plus also. So those are the two which we can we can work with, but we don't synthesize lipids ourselves.
If I can add a point, just keep in mind that at Tebubio, in terms of supplier of lipids, we have many of them. Xavier have already introduced some: Polyplus, Certest, Echelon, Broadfarm. Keep in mind that for our laboratory services, we can use all of them for your project. And Xavier and Erika will select the best lipid to match your needs and to find the best formulation. So that's really something you have to keep in mind. We can use all of the lipids of our supplier for our laboratory project. Okay, a new question now, more on the case studies. The LNP giving 63% of encapsulation efficiency, but work very well. It could be related to the limit of the use test for the analysis of encapsulation efficiency.
I'm not sure I understand the question, sorry. So could be related to use.
Maybe that's...
I don't think so. I mean, no, it might be the other way around, meaning, for example, if you dilute too much your LNPs before doing the ribogreen assay, then you wouldn't see, for example, free mRNA. Well, mRNA that is not encapsulated in the LNP. So here, I don't think so. Maybe it's because there was too much mRNA for the amount of lipids that we had, or maybe we had to tweak a bit the lipid proportion for it to work. But I don't think so. I don't think it was a bias through ribogreen assay.
Okay. Maybe Katia, if you would like to add more clarification or on your question, I'll let you ask a new question in the chat. Okay, next question. I have a question on the size of the encapsulated mRNA. Do you experience or expect a big difference in terms of encapsulation efficiency for small mRNA of about 50 to 100 nucleotides?
I don't think so. No, you have to optimize, but as I mentioned, the LNPs will just accommodate around your mRNA. So it's not like it's fixed size and then it goes into it. So it will just, go around your mRNAs. And with the microfluidics, you can really, if you increase the total flow rate, you can also decrease the particle size. So I don't think so.
Yeah, that's okay, Xavier?
Yeah, no, I was just,
Yeah.
sorry, I just spaced out. No, I was thinking.
Perfect. New question. How did you check the poly A size?
So the poly A size, so it's encoded in our plasmids. So basically it's fixed. So we know that it's 120 because that's, and we have QCs to confirm that on the QC on the plasmid. We haven't sequenced the mRNA, if it's the question.
Yeah, basically we don't check the size of the poly-A tail in the RNA, but we check it in the plasmid that is used as a template. And so, as Xavier was mentioning during the webinar, we have developed internally an optimized DNA template for efficient transcription and translation in mammalian cells. And this one has a poly-A tail of 128, which is fragmented to increase the stability, especially against recombination in bacteria during the plasmid preparation, which is a known issue for long poly-A tails. And what we have is during indeed the plasmid preparation is what we call a poly-A warranty, meaning that at the moment of cloning and amplification of the plasmid, several clones are screened until the right poly-A tail length is identified. So this way, basically, you can make sure that like your mRNA produced in different batches are reproducible. They always have the same poly-A tail length. And also, if, for example, you compare mRNAs of mutants, but you know that you're not going to have differences related to a different poly-A tail.
Okay.
Yeah.
Thanks, Xavier and Rika, for your answer. I really would like to thank you all for all your very interesting questions for our expert. A new one, okay. I have a question regarding the endocytosis, which you mentioned that it does not correlate to protein expression, as evident from one of your formulation. Is it due to poor endosomal release? And have you tried to find the reason behind this phenomenon?
That's a very good question. The answer is no, we haven't tried, but because of lack of time, but that would be really, because that's an hypothesis. I don't know. Well, it seems that there is either sticking to the membrane or membrane fusion, but we haven't checked that. So either there's a problem during endocytosis, or as you said, during, which is to me most likely, but maybe not, I'm not sure, most related to endosomal escape, but we haven't checked that, but that would be very interesting.
Thank you, Xavier. Have you used any other method to test encapsulation efficiency apart from ribogrinase?
Not really. I did some tests, but not very conclusive, using just UV and trying to isolate mRNAs that way, but that didn't work very well. So for now, I mean, and ribogrin works quite well.
Thank you, Xavier. A new one. Did you check the mRNA degradation after the LNP synthesis?
For this one, no, but we did, yes, and it's fine. This is something that we can do as well. We even tested after as a QC after, I don't know, maybe 2 weeks in the fridge just to see how the mRNA looked, and it looked fine. So yeah, but not for this particular project, no.
A new question. You have mentioned many hurdles in achieving efficient LNP preparation, but will this be sufficient to circumvent the complexities of the in vivo system and avoid off-target effects? As we know, endocytosis is a bottleneck for LNP translation. What solution do you propose? Have the optimization you implemented demonstrated a distant profile compared to what is already known about these lipids?
That's a very, very difficult question. So we don't do in vivo first and foremost. So we rely on data from our suppliers and partners, from the literature for that. We have collaborators that can do it. I don't think, to be honest with you, I don't think we have revolutionized the targeting LNPs. It's still quite open. There's a lot of things that can be done that are done right now with the conjugated LNPs, and I agree that targeting remains an issue. And even if targeting the amount and the percentage of mRNA actually endocytosed, is very low, but we are not, maybe that wasn't clear, we don't develop ourselves LNPs. So we just work with trusted partners and suppliers that really are experts on that field, and we use this expertise to really help customers circumvent some bottlenecks.
Thank you, Xavier.
I hope I answered the question.
If you need more detail, you have two options. You can ask in the Q&A box, or you can in the survey you can write your question, and Xavier will come back to you to discuss a little bit more into the detail about the point you would like to check. New question. I think that's linked to the method used with encapsulation efficiency. So how it get encapsulated in your method? What is the mechanism?
So you mean how we calculate the encapsulation efficiency, basically? That's the question?
No, I think it's just a microfluidic system.
Yeah, I think that's more how the mRNA is encapsulated in your method.
Okay,
I think this question is linked to the one who asked the type of solution you use to evaluate the encapsulation efficiency percentage. Now it's more the method you use for the encapsulation.
Oh, I apologize. So we use microfluidic systems. So the Spark has a the circuits are a bit like circles to mix the mRNA and the lipids, and for the Tamara, I think it's herringbone microfluidic system. Oh, we have also access to the baffle system. So these just dictate some properties regarding sizes of an LNP. So we use the microfluidic system to prepare the LNPs.
Okay, are there LNP formulation that target brain uptake from blood preferentially in mice, for example?
Not that I know of, at least not from, I know that some of our partners are working on that, but it's not commercially available yet. And from the literature, I'm unsure, to be honest with you.
Yeah, I would say also for this question, I invite you to fill the form at the end of the webinar, and we can discuss with our partners and see if they can propose something.
We can look into that, into data. Yeah.
Okay, thank you. New question. What is the approximative cost of the screening study with the 15 formulation? That's a good question.
That's a good question. I actually forgot. Yeah, I can, if you want more, we can discuss that later on, because honestly, I forgot. But we can make discounts.
We will come back directly after the event. New question. Do you know what's the mechanism by which LNPs have adjuvant effect?
Not completely, because I'm a biochemist, so all these sort of stuff eludes me. But from what I read, there is recognition by the immune system of the LNPs as foreign agents, so you can have antibodies directed against PEG and against some phospholipids. So maybe it's because of that, and I think it depends on the dose and how frequently, sorry, you inject stuff. But aside of that, maybe Erika, you.
Yeah, no, we should know the answer, but yeah, no, we definitely have to look more into it. But I see that it's actually one of our clients already who is asking the question, so we'll get back to you.
Okay. New question. Thanks for the presentation. Do you know how to QC the number of mRNA molecule encapsulated in each nanoparticle?
That's a very good question. No, maybe I'm not sure it would work. NTA, I was going to say probably like some sort of single cell analysis, but maybe NTA, but I'm not sure. That's a very good question. We know that on average, it's 1 to 2 molecules of LNP, of mRNA per LNP, but I guess that probably depends quite a bit. But we can also look into it, and if you're interested, we can, provide you some literature on that.
Okay, thank you. New question. Are all lipid nano LNPs, have the ability to escape the endosome to deliver their cargo into the cytosol, or to make them efficient to deliver to the cytosol?
Well, that's what a lot of people are doing. So mainly is, from what I understood, is from the ionizable lipid, because, so not to go into details, but it has to do with the change in membrane shape. Basically, if you look at the ionizable lipid, you will see that from a geometrical point of view, they look like a cone. Basically, the head is very small and the tail are very big. And this is because it has been known for decades now that lipids like that, so for example, phosphatidylethanamine, POPE, is known to do that. So those kind of lipids, they tend to form what is called hexagonal phase. And hexagonal phase is a bit like inverted micelles, meaning that instead of having the polaroid group around the LNPs, it's inside and then the tail outside. So they favor that, and because of this unusual structure, they sort of disrupt the membrane and it allows endosomal escape. So there's a lot of work going on into that to design ionizable lipid that enhance this effect. And there's probably other effects that I'm not aware of, but this is the part that I'm more interested in. So that's the one I know a bit more.
Thank you, Xavier, for the explanation. I think that's not a question, but that's more a comment regarding the previous question from the mRNA could be accessible with ribogreen probe if it's exposed on the LNP interface, for example, and documented in different publications. In that case, more tests should be added to determine if mRNA is really free or just accessible by the ribogreen probe.
Okay, I see. Yeah, that's a very good point. I haven't thought about that, but yeah, that's nothing to say. Yeah, that's a very good point.
Okay, next question. Did you check the plasmid size in the RNA or only on the plasmid? Poly-A.
Okay.
Oh, no, it's only on the plasmid.
Oh yeah, okay. So did you check the size of the poly-A tail in the RNA or only on the plasmid? Thank you for the precision.
So we do a QC regarding the size of the mRNA, but it's rough idea. So it matches what we expect, but maybe there could be some variation. That's true.
Okay, perfect. Another question, but finally you answered the question in another one. The question was, are you able to do screening in vivo model in addition to in vitro model? So yes, thanks to our supplier. And we will take the last, I think we'll take the last question. Thanks again for all the questions you have asked to our experts. So after the Spark preparation, what dilution factor do you use and what strategy do you use to prevent LNP fusion during subsequent concentration step?
So sorry, can you repeat the question one more time?
After the Spark.
So we do 3-time dilution, then we do dialysis, yeah, we don't really do concentration step unless it's asked, because we know, I mean, I worked a lot on proteins, and I know when you concentrate, you tend to lose quite a bit of material. Well, it depends, but we prefer to avoid it unless we haven't had the choice. But otherwise, we don't really add anything to prevent fusion. I guess you could add some, yeah, could add some stuff, but no, we haven't checked that actually. But we did actually, sorry, I remember one thing. I did some tests regarding concentration, and I used the regular QC, and I couldn't see much difference in particle size actually after concentrating. So I didn't see much of fusion, because yeah, I would expect size to increase if there was some fusion or increase in polydispersity, which was not the case here. And we used it was in PBS, so maybe just the salts were enough in our case. Not sure.
Perfect, Xavier. Thanks, Xavier and Rika, for your answer. Again, thanks all for all your questions. I still have 2 minutes to conclude this very interesting session. So just before to end the session, I would like to remind you about our upcoming webinar next month, co-organized with our partner CETest, where we'll explore their in vivo delivery technology and solution. Maybe for the one who asked in vivo screening, that should be that's a very interesting session to attend. Also, please take a moment to complete the survey that will appear in your browser when you will leave the session, to share your feedback, all your questions. If you need more, if you have questions, will come. If you need more or precise feedback, don't hesitate to ask all your questions in the survey, and let us know if you would like, of course, more information. Okay, I would like to thank you all for your attention and for joining today's webinar. I also would like to thank Xavier and Erika, our two experts, for the presentation and the question and the answer. So we hope the information shared has been helpful and very helpful and inspiring. So please feel free to reach out if you have any further question or would like to discuss how we can collaborate together for your delivery project. So thanks again all for your attention. Thanks Xavier, thank Erika, and hope to see you next month in our next delivery series webinar. Thank you all.
Here.
Thank you. Bye.
Bye.
Bye.